Hi everyone, I'm Monica Grabovitz and my topic is on designer babies. What's a designer baby? A designer baby is a baby genetically engineered in vitro for specially selected traits, which can vary from lower disease risk to gender selection. And these traits can include sex, disease, athletic ability, hair or eye color, personality. The first designer baby was in 1990 named Adam Nash. Scientists selected his embryo so that he would have the right cells to save the life of his dying sister. His sister suffers from Fanconi's anemia, an autosomal recessive disease that affects bone marrow, which results in decreased production of all types of blood cells. Once he was born, he became a donor to his sister, and there was a blood transfusion, which has more than doubled her survival rate. In vitro fertilization, or IVF, has become an increasingly common procedure that helps couples with infertility problems conceive children, and the practice of IVF confers the ability to pre-select embryos before implantation. Scientists have also reported a method of extracting defective mitochondria, the energy powerhouses of cells, from a woman's egg and replacing them with a healthy mitochondria from a donor egg. The CRISPR-Cas9 system is currently the simplest, most precise method of genetic manipulation that allows researchers to edit parts of the genome by removing, adding, or even altering sections of the DNA sequence. Its precision can alter DNA down to the level of a single letter. By editing the DNA of these cells or the embryo itself, it could be possible to correct disease genes and pass those genetic fixes onto future generations. How does the CRISPR-Cas9 system work? The Cas9 protein forms a complex with guide RNA in a cell. This complex attaches to a matching genomic DNA sequence adjacent to a spacer. Then the Cas9 RNA complex cuts the double strands of the DNA, in which lastly programmed DNA may be inserted at the cut. CRISPR has many applications that includes correcting genetic defects, treating and preventing the spread of diseases, and also improving crops. The method to create a designer baby is pre-implantation genetic diagnosis, or PGD. This allows viable embryos to be screened for various genetic traits, such as sex-linked diseases, before implanting them in the mother. Through PGD, physicians can select embryos that are not predisposed to certain genetic conditions. For this reason, PGD is commonly used in medicine when parents carry genes that place their children at risk for serious diseases such as cystic fibrosis or sickle cell anemia, as well as Down syndrome. There are a few steps for PGD. The first step is that a few cells are microsurgically removed from the embryos, which are about 5 days developed, and after this collection, the embryos are safely frozen. The DNA of cells is then evaluated to determine if inheritance of a problematic gene is present in each embryo. This process takes usually about 1 full week. Once PGD has identified embryos free of genetic problems, the embryos will be placed in the uterus by IVF. And then the last is that they usually just wait for implantation in a positive pregnancy test. Fertilization, along with PGD and a successive frozen embryo transfer, it usually costs about $17,000 to $25,000 in the United States. Many countries completely prohibit PGD, and that includes Austria, Germany, Ireland, and Switzerland. And then other countries restrict PGD, but only to medical use, and that includes France, Greece, Italy, and Norway as well. However, the United States federal law does not have any regulation on PGD, but those who are in favor of PGD believe that the government should not be involved in the procedure and that parents should have their own reproductive choice. There are many pros and cons to having a designer baby through PGD. The pros are that it reduces risks of genetic diseases, it reduces risk of inherited medical condition, and it can give child genes that parents don't carry, and they prevent the next generation of family from getting characteristics and diseases such as Alzheimer's, Down syndrome, or cystic fibrosis. PGD also can test more than 100 different genetic conditions, and this procedure is performed before implantation, which allows the couple to decide if they wish to continue with the pregnancy. The procedure enables couples to purse biological children who might not have been done so otherwise. A few cons of PGD includes termination of embryos, possibility of damage to the gene pool, baby has no choice in the matter, and it has loss of individuality. There is a major ethical deliberation on experimenting with fertilized eggs in embryos and killing the flawed ones. As of 2017, the United States restricts the use of these procedures, and it's under heavy regulation by the FDA and NIH. Finally, designer babies represent great potential in the field of medicine and research, but there still remains many ethical questions that need to be addressed. Many ethical questions about this method. The majority of people do like that there are more pros, that they can pick and choose characteristics of their babies, which can also increase their lifespan, prevent future generations from undesired diseases and characteristics. They can modify the gender, the physical appearance, the level of intelligence, personality traits, and they can know their exact child's genome, which can improve their lifestyle. Although currently you can reduce the certain disabilities in a designer baby, you can't right now choose the cosmetic and personality traits, but researchers do believe within the next few years they'll be able to do that. Any questions?